Australia into the wilds of the Kimberley. It's been disastrous for local wildlife, but local rangers are taking on the toads with the help of...
For 90 years, Australia could not win. An army of poisonous invaders was rolling across the continent's north, and every weapon the country threw at it bounced off. Then a small team of biologists noticed something strange happening in the mud at the edge of a billabong, and what they found there would eventually turn the most successful invasion in Australian history into the hunted. This is the story of how the continent's deadliest invader met a predator it never saw coming. And it started with a tin of cat food. Let's go back to where it began. It's 1935 and Queensland's sugar industry is in trouble. Greyback cane beetles are tearing through the root systems of sugarcane fields, and nothing the Bureau of Sugar Experiment Stations has tried is working. Then word arrives from a sugar industry conference that a large South American toad had apparently helped control beetles in Puerto Rico and Hawaii. An entomologist named Reginald Montgomery travels to Hawaii, collects a breeding population, and brings just over 100 toads back to a research station near Gordonvale, North Queensland. By August of that year, those 100-odd toads have bred in captivity, and 2,400 of their offspring are released into the cane fields. Nobody has tested whether the toads actually eat the beetles. Nobody studies what else might happen if they don't. The release goes ahead anyway, it doesn't work. The beetle larvae live underground and high on the cane stalks, completely out of reach for a toad that hunts on the ground. The entire justification for bringing them to Australia evaporates within a season. But the toads don't care. They've landed somewhere warm, wet, and packed with prey that has never had to deal with anything like them. Insects, frogs, small reptiles, anything that fits in their mouth becomes food. And then the part nobody saw coming. A single female cane toad can lay tens of thousands of eggs in one season, and she can breed more than once a year. With no native predator able to safely eat an adult toad, the population doesn't grow steadily, it explodes. Within a few decades, the descendants of that one Queensland release have pushed across the entire top of the continent, through the Northern Territory and into the Kimberley region of Western Australia. And the invasion wasn't just spreading, it was accelerating. Researchers tracking the front found that the toad's rate of advance increased roughly fivefold since the 1930s, from around 10 kilometers a year to over 50. The toads doing the leading were measurably different from the ones following behind, longer-legged, more enduring, faster. Evolution was actively building better invaders out of whichever individuals reached new ground first, and their offspring inherited the advantage. The toads weren't just spreading, they were being upgraded by their own conquest generation after generation. What made them so deadly wasn't speed though, it was chemistry. Cane toads carry parotid glands packed with a toxin called bufotoxin, a compound that disrupts the sodium-potassium pumps that keep a heart beating. Any predator that bites down and swallows gets a dose strong enough to stop a heart within minutes. Australia's snakes, monitor lizards. And quolls had spent millions of years learning how to handle local prey, and none of it prepared them for this. The northern quoll, a fierce, cat-sized marsupial hunter, is now listed as endangered under Australian law specifically because of cane toad predation pressure, according to Western Australia's Department of Biodiversity, Conservation and Attractions. Goannas and freshwater crocodiles have suffered the same fate in toad-affected regions. When a toad front rolls through an area, the established predators that try to eat it start dying within the same season, and everything below them in the food chain shifts because the things that used to keep it in check are gone. For decades, Australia threw almost everything it had at the problem. Mass collection drives, physical barriers, chemical research, even genetic approaches. None of it scaled. You cannot manually remove a population spread across an entire continent and numbering in the hundreds of millions. The toads just kept coming, and every year they came a little faster. If you want to find out what finally turned the tide, stick around, because the breakthrough wasn't a weapon humans built, it was something that had been living alongside the invasion the entire time, and nobody had thought to look at it properly until they did. Here's where the story turns. A research team led by Professor Rick Shine at the University of Sydney, with biologist Georgia Ward-Fear as a key investigator, started watching what happens at the water's edge in the days right after cane toad tadpoles finish metamorphosis. That's the moment a tadpole loses its gills, grows legs, and crawls out onto land as a tiny toad for the first time. And the team noticed that something was already attacking these baby toads in large numbers, a native insect called the meat ant, scientific name Iridomyrmex rubarus. The question that mattered wasn't just that the ants were attacking, it was why the ants didn't seem to care about the toxin at all. The answer comes down to anatomy. Bufotoxin is built to disable the cardiovascular systems of vertebrates, animals with hearts that run on the same basic electrical wiring, whether you're a snake, a lizard, or a crocodile. An ant's body doesn't work that way. Insects are far less sensitive to this class of toxin than vertebrates are, which means a chemical defense fine-tuned over millions of years against snakes and birds in South America is essentially invisible to an Australian ant. The meat ant doesn't avoid the toad's poison, it simply isn't built to register it as a threat in the first place. And meat ants are not a passive species. They're large, fast, aggressive foragers that hunt in coordinated numbers. A single scout finds a target, lays a chemical trail back to the colony, and reinforcements arrive within minutes. When a colony encounters a freshly metamorphosed toad, slow, small, and with no working escape response, it isn't treated as a threat, it's treated as food. Published research from Shine and Wardfear's team in the journal Functional Ecology ran direct comparisons between cane toad metamorphs and 7 species of native Australian frogs raised in the same enclosures. Across nearly every measure of escape behavior, the toads performed worse. The native frogs reacted to an approaching ant with a single decisive jump that broke contact immediately. The toad metamorphs were more likely to stay in the open, stay active during the day when ants are most active, and freeze instead of fleeing. The researchers described this mismatch as an evolutionary trap. A trait that worked perfectly well in the environment that toad evolved in becomes a fatal liability in an environment it never adapted to. There's also a numbers problem working against the toad. A predator that bites a toad and swallows it gets a concentrated, often lethal dose of toxin through the gut. A meat ant biting the skin at a single point gets a trace amount nowhere near enough to matter, and it doesn't need to swallow anything to do damage. The toad's entire chemical defense was built around the assumption that something dangerous would try to eat it whole. Ants don't do that. They attack from multiple points simultaneously and overwhelm through sheer numbers, and the toxin coating the toad's skin does nothing to stop it. This is where the science turns from observation into a strategy. If meat ants were already killing toad metamorphs without any human help, the obvious next question was whether their numbers and timing could be deliberately amplified at the exact pinch point in the toad's life cycle where it's most vulnerable. Newly metamorphosed cane toads face a brutal set of constraints all at once. They've lost their gills, so they have to leave the water, but their skin hasn't toughened enough to handle dry conditions, so they're stuck in a narrow strip of damp ground right at the waterline. Adult cane toads are cannibalistic toward their own young, so the metamorphs are pushed to move during the day when adult activity near the water is lower, which also happens to be when meat ants are most active. Every pressure on the baby toad is funneling it toward the exact place and the exact time of day where its one undefeated predator is waiting. Researchers tested whether they could exploit this by baiting shorelines with simple protein bait, tins of cat food, to draw meat ant colonies toward ponds just ahead of a predicted metamorph emergence. According to the team's published findings in the Journal of Applied Ecology, adding bait at these sites increased meat ant density at the shoreline more than 4-fold. And over half of the ant attacks recorded on metamorph toads in the field were immediately fatal. Most of the toads that initially escaped an attack didn't survive long afterward either. The same study found that roughly 88% of those that got away still died from their injuries within a day. This wasn't a marginal improvement. It was a near total kill rate concentrated at a single point in the toad's life cycle, using nothing more than food a colony of native ants was already wired to want. Other published work from the same research group looking at the physical terrain along these pond margins added another layer to the picture. Much of the top end and Kimberley shoreline has been churned up by cattle and other livestock hooves, leaving a landscape of small craters and ridges. To a human, that's rough ground. To a toad metamorph barely the size of a fingernail, a single hoofprint can be an inescapable pit. The research found that steep-sided hoofprints increased the toad's exposure to ant attack and to drowning after rain, while native frogs sharing the same ponds showed no comparable spike in mortality because their escape instincts were already built for this environment. That last point matters enormously, and it's worth sitting with before celebrating anything. Meat ants are native to Australia. They've coexisted with native frogs and the rest of the ecosystem for a very long time, and the research consistently shows that native amphibians aren't being harmed at anywhere near the rate cane toads are at these baited sites. That's the whole logic behind using this approach instead of yet another imported fix. You're not introducing something new and hoping it behaves. You're concentrating a predator-prey relationship that already exists in nature at the specific time and place where it does the most damage to the invader and the least damage to everything else. But scientists involved in this line of research have also been careful in their published papers to frame it as one tool within a broader, multi-pronged approach to managing cane toads, not a single silver bullet that ends the problem outright. Concentrating any species artificially above its natural density, even a native one, requires monitoring. Because ecological side effects are exactly the kind of thing that got Australia into this mess with the toad in the first place. The strategy researchers describe is targeted, known cane toad breeding ponds, baited during the specific window of peak metamorph emergence, monitored before and after, then left alone for the rest of the year so ant colonies return to their normal numbers. That's the part that makes this approach so different from everything that came before it. The 1935 mistake was built on adding something new to the system and hoping for the best without testing whether it would even work. This approach starts from the opposite instinct. Find what the ecosystem is already doing and give it a push at exactly the moment that push matters most. Cane toads have now spread across a huge portion of tropical and subtropical Australia, and government agencies are direct about the limits of what's achievable. The Department of Climate Change, Energy, the Environment and Water states plainly that there is no broad-scale way to eliminate cane toads across the country. Full eradication isn't the goal of this research, and no scientist working on it claims otherwise. The goal is suppression at key points, breeding ponds where targeted intervention can meaningfully cut the number of toads that survive to adulthood and start the egg-laying cycle all over again. And the math behind that is worth taking seriously. Every female that doesn't reach adulthood is not just one fewer toad. It's tens of thousands of eggs per breeding season that never get laid, compounding across every future generation she would have produced. A method that can eliminate a large share of a season's metamorphs at a single pond, repeated across multiple high-value sites and multiple breeding seasons, has the potential to meaningfully slow the population growth that has driven this invasion for 9 decades, even without ever reaching zero. Northern quail populations and monitor lizards have been documented recovering in some areas where toad pressure has eased over time, a sign that the food web disrupted by the original invasion isn't necessarily gone for good, given the right conditions and enough time. None of this is presented in the research as a finished victory. It's presented as evidence that the right kind of pressure, applied at the right moment, can bend a curve that for 90 years looked unbendable. What's remarkable about this whole story is what it reveals about the limits of the toad's famous defense. Bufatoxin made the cane toad nearly untouchable against everything it evolved to face in South America: snakes that swallow, birds that grab, mammals that bite. It was such a complete defense against that specific category of threat that Australia spent decades assuming there was no answer at all. What the meat ant research exposed is that the toad's defense was never universal. It was a perfect answer to one kind of attack and no answer whatsoever to a different kind, a coordinated day active. External swarm attack from an insect too small for a single dose of poison to stop and too numerous to fight off one at a time. The toad brought the only weapon it had ever needed, and for the first time in 90 years it ran into something that weapon simply doesn't work against. The breakthrough here didn't come from a laboratory inventing something new. It came from scientists noticing a predator-prey relationship that Australia already had, sitting right at the water's edge the entire time, and asking how to give it a more decisive advantage at the one moment it mattered most. No expensive infrastructure, no chemical contamination, no new species introduced into an already damaged system. Just bait, timing, and attention to what the ecosystem was already trying to do. There will be ponds the program doesn't reach in time and cohorts of toads that slip through regardless. The cane toad is too deeply established across too much of the continent for any single method to undo 90 years of spread quickly. But for the first time since 1935, researchers have a tool that targets the invader specifically at its most fragile life stage, built from a relationship the ecosystem had already worked out on its own. In a fight Australia had been steadily losing for 9 decades, that shift in direction is what counts as a turning point.